In this lecture, we're going to focus on what the scientific method is and how it relates to chemistry. As you know, chemistry is the science that studies the materials of the universe and the changes that these materials undergo. So what a chemist will do and what you will study is a connection between the macroscopic world you live in, macroscopic meaning large scale, in other words, a big chunk of gold, a bone, a lake, a river, so large things, and the microscopic world that makes everything work. Microscopic means on the level of elements and molecules and atoms, protons, electrons, and neutrons, and how they interact with each other. So chemists look inside of living and non-living things to study how their components behave. Here's a few examples of things that you will learn in your chemistry class, and each one of these shows a good example of how the macroscopic world is caused by changes in the microscopic world. We're going to focus most of this presentation on the scientific method and how to think like a scientist. As you can see from this diagram out of your textbook, the scientific method involves the process of making observations, analyzing, and trying to explain and manipulate the world around us. So if you make an observation, you will then make a hypothesis and then experiment. That will then get revised and you will try it again, and that can eventually become a theory or a law. And theories and laws can be modified as needed. The scientific method can be used to solve a problem or to achieve a certain outcome. For example, an engineer may build a device to try and solve a problem. A pharmaceutical company may develop a certain drug or medication to try and cure a disease. So both of those examples involve use of the scientific method. You've actually been using the scientific method your whole life. For example, learning how to walk and to feed yourself, you had to use the scientific method, and figuring out the fastest way to get from my classroom in room 112 over to the cafeteria. For example, my daughter, when she was a toddler, she was very good at using the scientific method. She would see the VCR, and when she wanted to watch a Disney movie, she would see that we would take that movie and we would put it in the VCR. So for example, her favorite Buzz Lightyear movie. So here's her little eyes viewing that VCR. She then thought about other things she could put in the VCR, such as her teddy bear, a bottle, the ball, and she kind of developed ideas and observations. She tried the ball, it didn't go in. She made that observation. She tried the teddy bear, it also did not fit in. So she realized that the ball shape and the big smushy shape did not work. Eventually, over the course of a few months, she realized that there was a long rectangular shape, and that is what made the VHS tapes fit in. And so she started looking for things that were that, and by the time she was about 2 years old, she figured out a sandwich would fit in to the VCR. So let's review each of these steps of the scientific method one step at a time. The first step is to gather information about the world around you. So we do that by making observations and inferences. The difference is that an observation is something you see, hear, feel, or smell. An inference is how your brain interprets that information. So for example, you may observe that the burner on the stovetop is red. However, you don't see that it is hot, you infer that it is hot. So unless you have infrared vision, which basically shows different temperatures as different heats or as different colors, you can't see with your eyes that it is hot. So the observation is purely what you witness with your five senses. The inference is then what your brain thinks about it. Step 2 is to come up with a plan. Once you've made many observations, you might be able to find a possible cause and effect relationship in what you see. There are many variables that you're going to test when you're looking at having an experiment, and the first variable is the one that is the cause in the cause and effect. That is called the manipulated variable. To manipulate means to change or to do things with your hands, and so the manipulated variable is the factor that is changed by the scientist. It is a factor in an experiment that causes the results to occur. It is the variable that is controlled and changed by the scientist. This is sometimes also known as an independent variable, and in a controlled experiment there can only be one manipulated variable. The second type of variable is the one that is the effect. This is known as the responding variable because this variable responds to changes in the manipulated variable. So the responding variable is the factor in an experiment that responds to the manipulated variable, otherwise known as the effect. This variable is also called the dependent variable, and there may be more than one responding variable in a controlled experiment. Oftentimes when you are analyzing a particular problem, however, you're going to pick one to focus on. Every other item in your experiment is going to be a controlled variable. These variables are the different factors that are kept identically the same from one trial to the next. For example, any equipment or glassware you use, you want to keep the same. The conditions of the laboratory or classroom, the temperature of your solutions, anything that is not a variable that you want to be changing, you have to keep it the same to keep your experiment controlled. The third step is to make an educated prediction. People often think of a hypothesis as an educated guess, but we don't want to make it a guess. A guess is just random. A prediction is actually based on knowledge, so you're trying to predict what will happen based on previous knowledge. A hypothesis is a testable explanation for what you have observed, and it must include three things. Usually, we science teachers have students learn if, then, because as a hypothesis statement. What we're really trying to get you to write down, however, is the relationship, the prediction, and the explanation of what's going to be going on in your experiment. So first you're going to state the relationship between the manipulated variable and the responding variable. Secondly, you want to include your prediction of how the variable will respond. And finally, you want to include an explanation of why the predicted outcome will occur. If my daughter were to have written down a hypothesis for her sandwich in the VCR, it might have something along the lines of, if the sandwich is a rectangular object and the VCR opening is rectangular, then the sandwich will fit into the sandwich will fit into the VCR because a rectangle will fit into a rectangular spot. Something like that. That's obviously not very smooth, but that's what we're looking for with an if, then, because statement. And a hypothesis does not necessarily have to include the words if, then, because, but it does need to convey the relationship, the prediction, and the explanation of what you're intending for your experiment. Step 4 is to test your hypothesis. In a controlled experiment, as we mentioned, there is a test where there's only one variable, one manipulated variable being changed to see if the outcome changes or not. So as part of your controlled experiment, you're going to have data and an analysis. There are two types of data. The first type of data refers to quantities, and it is called quantitative data. So note the N in this statement. A quantity is anything that is being measured or counted. For example, change in temperature, pH, grams, volume, those would all be quantities that are measured. The second type of data is qualitative, and that involves different qualities or descriptions of your substances. And note the L in this one that sets it apart. So this could be changes in color, changes in smell, changes in appearance, cloudy versus transparent. That would be qualitative data. Finally, you would have an analysis section in which you compare the data in each trial of your experiment to analyze your results. Step 5 is to make a conclusion about whether your hypothesis was supported or not. It's important to know that a hypothesis is never considered to be right or wrong. It's considered to be supported or not supported by the data. So, if the data does not support the prediction you made in your hypothesis, then you can do one of two things. You can either make a different variable or choose a different variable that you can test to see if that makes the hypothesis more accurate, or you can change your hypothesis. You can revise it and then test that. However, if the data does support your prediction from your hypothesis, you should definitely repeat the experiment just to make sure it is correct. Finally, if the data does support the prediction, it's always interesting to change some of the variables to see under what conditions the hypothesis is true. When a set of variables have been tested repeatedly, they can sometimes lead to the development of a theory or a law. So after you have revised and retested your experiment, a tested explanation of an observation or a hypothesis that has been tested and supported many times can develop into a theory. A theory is an explanation of something that is observed. So a theory will never become a law. Theories and laws, as you can see from the diagram, are two different outcomes of the scientific method model. They do not turn into one another. A law, by contrast, is when something is observed many times and gets observed the same way. So an observation may lead into a law, while a hypothesis may develop into a theory. They're sort of two separate paths that can come out of the scientific method, and those two different paths or roads, they do not run into each other. With repeated experimentation and different changing the variables and conditions, a theory and a law can be changed over time, as you can see in the diagram here. When new data is uncovered that opposes the theory or law as it is currently written, then that theory or law can be modified to fit the current conditions in the modern day. Finally, as a scientist, you will definitely want to share your results with others so that you can be published and hopefully get lots of grant money for your laboratory and become world famous and have something named after you. So when you're sharing your results, you need to have an excellent data table and a graph. A data table is a table that organizes all of the qualitative and quantitative data from your experiment. It must include a title that describes the data and headings for any columns, and those headings must include the units of any measurements. So if you're measuring changes in volume, you must include if that volume was milliliters, centimeters cubed, etc. And most importantly, for when you share your results and publish your findings, you need to have an excellent graph that makes it easy for the public to understand how your data shows the relationship between the manipulated and dependent variable. So your graph must have a title that describes the two variables and the relationship. And the graph should be in what's called a dry mix, or dependent on y, manipulated variable on x form. So in orienting your graph on the y-axis going up and down, you would want to have the dependent, otherwise known as responding variable. And on the x-axis going horizontally, that's where you would include the manipulated, otherwise known as independent variable. So dry mix will help you remember that. Finally, you need to label your axes, and again, the units of any measurements must be included on those axes. Hopefully this lecture has given you a better understanding of the scientific method, and at least a better understanding of the notes as they apply to our quiz this week.